Thank you all for the opportunity to speak to you today. My name is Jeff Dodd, and we'll be talking about our findings of how obesity may be affecting ischemia reperfusion injury in the lung. We use a mouse model for this. You can see there's lots of people involved in this. I happen to be the spokesman. I don't have any competing interests. So our basic hypothesis was that obesity acts as an inflammatory state. And what happens is that the fat cells recruit macrophages and activate them to be inflamed. This then exposes the body to a continued inflammatory burden, and the reserves to combat a subsequent injury then are reduced. There's lots of indications in the literature of two-hit models that use the same concept. Some of them are from us, some of them are from others. But we did feel this was a type of a two-hit model. So what we did was we took mice and we fed them either normal chow or high-fat diet for 18 weeks. At the end of 18 weeks, we did find that the high-fat mice did gain more weight. At the end of this 18-week period, then we would, through a thoracotomy, expose the left pulmonary artery and put a tie on it so that we would cause ischemia. The bronchus was left patent, but the pulmonary artery was occluded. While the pulmonary artery was occluded, we intravenously injected Evans blue dye. Evans blue dye binds albumin, so anywhere albumin went, the tissue would turn more blue than if it didn't go there. At the end of a 30-minute ischemic period, we would reperfuse. We would pull the tie, allow it to reperfuse for 150 minutes. Then we would check the lungs to see if the left lung versus the right lung had more blue in it. To measure blue, we did a ratio of absorbance for the Evans blue, and we found that a higher number is worse, a lower number is better. In the normal diet mice, we do actually have an ischemia reperfusion model, so we do have more leak from the left lung than we do with the right lung. And with the high-fat diet mice, as opposed to the normal chow mice, this injury is worse. We then took these mice, and after 18 weeks, continued to feed them either their normal diet or their diet with liraglutide and vitagliptin, a GLP-1 agonist, DPP-4 antagonist. And we found, again, this is what we found earlier, just a repeat of that, we found that high-fat diet is worse than normal chow. If you give the blockers to a normal chow diet mouse, there was no real difference, but if you give it to a high-fat diet mice, you reduce the injury it otherwise had. And associated with that, we did find some inflammatory markers went down. So we looked in the blood and found that IL-17, MCP-1, and interleukin-1 beta, the normal chow looked like this, and the high-fat diet mice, they all went up. And then when we get the drugs, they went down again. Then when we looked at the macrophages to see whether this was consistent, we found that some macrophage numbers actually did go up, but not all of them. So compared to normal chow, the alveolar and the LY6C negative macrophages did statistically increase in number, but the interstitial macrophages did not increase, nor did the LY66 positive ones. Then we looked at the inflammatory state of these macrophages, and we used a volcano plot to do this. So using a volcano plot, on the x-axis it tells you the magnitude of change. So in a log base 2, a value of 1 means a twofold increase, and a value of -1 means a 50% decrease. To look to see whether those changes were statistically significant, you look on the y-axis, log base 10, a value of 1.3 is equal to 0.05, a value of 2 is equal to 0.01, value of 4 is equal to 0.001. And if we look back at our macrophages, again, there weren't a lot of inflammatory markers changed with the alveolar or the interstitial macrophages. There were some with the LY6C negative and LY6C positive. As you can see, Indicators of the co-stimulatory pathway are increased. Indicators of the complement pathway are increased. Some anti-inflammatory markers are decreased, but again, it's not really 100% consistent. So this was a little less exciting to us. I wonder if something else is going on. Could this be affecting the energetics of the cells? So we then looked at AMPK level, and we found that in fact, high-fat diet mice had lower AMPK levels than the normal chow mice. We thought, I wonder why this is happening. How could the high-fat diet be signaling this? Well, there are ways that cells communicate with something that are exosomes. And exosomes are little vesicles that contain snippets of DNA. They're not necessarily snippets of RNA. They're not necessarily doing anything other than causing an increased read or a decreased read of otherwise present RNA, but they can modify the RNA reading. And so we wanted to see, can we find these exosomes, and are they increased perhaps in mice that are high-fat diet mice? So how do you do that? Well, what you do is you label exosomes, which happen to be indicated by CD63, with a marker that will light up. So if you look then, these are cells. This is the nucleus of the cell. This is sort of the membrane of the cell. If you can light up these markers, you'll be able to see them. So these big green dots then will be the exosome. Now, how do you make the exosome light up? Well, what you do is you take the DNA and you manipulate it. You manipulate it such that when you've come across the gene that codes for CD63, codes for the exosome, you attach to that a marker, a green fluorescent protein, which will light up. And this green fluorescent protein then will mark the exosome. The thing is you don't want all exosomes to light up. You just want exosomes from fat cells to light up. So you make the reading of this GFP conditional. It won't be read if there's this blocker in front of it. And you add to cells from which you want this blocker removed a bacteria that will eat up this blocker. That will remove this and allow the GFP to light up the exosomes only if they come from that cell type of interest, which is what we did. We had this conditional GFP read, and we had the bacteria within adipose cells. So the adipose cells, the bacteria would snip that away, allow the CD63 to be read, to the GFP to be read and light up the CD63, so you can see the CD63. It'll light up only in exosomes that are derived from fat cells and not those that are derived from other cells. So what happened when we did this? Well, this is what happened. This is the normal chow mice. These are the high-fat diet mice, and you can see all of these blue-green dots here. Those are all exosomes from fat cells. And those exosomes are statistically higher in the alveolus, and the bronchioles, and the blood vessel. And then we found that those exosomes actually contain LET7. LET7 has been shown to decrease reading of AMPK. So basically what happens then is that these exosomes carry something which decreases the reading of AMPK, which decreases the energetics within a cell. So in short then, what we find is that high-fat diet mice are sensitized to lung ischemia reperfusion injury. that this is likely influenced by exosomes that cause the downregulation of AMPK. The process seems to be influenced by the GLP-1 system. So again, I want to thank you for the opportunity to speak to you, and I'm open to questions if anybody has any.